Welcome back to this second video on the introduction to the respiratory system physiology. In the previous video, we were talking about the alveoli. And what are they? Well, these are pouch-like structures covering a surface of about 70-90 square meters, embedded in a mesh of capillaries. And the acinus is considered the functional unit of the lung and is composed by the respiratory bronchioles, the alveolar ducts, and the alveolar sacs. And at the end, on the terminal part of the alveolar sacs, you can find the alveoli. And as I said, the alveoli are embedded in a dense and thin mesh of capillaries that are coating the actual alveoli. Well, they are essential for the exchanges of gases and obviously important in the oxygenation of the blood. How does it happen? So the deoxygenated blood arrives at the respiratory bronchiole from the heart through the pulmonary artery, which becomes bronchial artery, shown in blue here in this picture. Now, is the blood contained in the pulmonary artery is saturated or contains carbon dioxide. That's why, although it's an artery, because it's a vessel that is coming out from the heart or going away from the heart, is often depicted in blue because it's containing blood with carbon dioxide. So the pulmonary artery or bronchial artery will divide in many capillaries that will coat the alveoli. Then the capillaries then will join back to form vessels, bronchial vein, which will become pulmonary vein in red in here, that are returning the blood full or charged with oxygen back to the heart. So although it's called vein because it's a vessel that goes toward the heart, is carrying blood that is full or charged with oxygen. From the standpoint of gas exchange, alveoli need to be as small as possible. Why? To increase the surface area to volume ratio. And how does it happen? Well, this is ensured by the presence of surface tension. This tension, this force, is generated by cohesive intermolecular forces in the liquid mass, so between the molecules of water, at liquid-gas interface. What does it mean? So there are intermolecular forces between the attractive forces between fluids that are coating the alveoli that are stronger than the attractive forces between the fluid and the gas itself. So these forces are strong. And because of that, there is the creation, they create a tension, which is a physical property that minimizes the surface area. This force, which is regulated by this Laplace law, is important because it's decreasing the radius of the alveolus. So again, so these attractive forces between the water molecules in the liquid film that lines the alveolus or all the alveoli are responsible for the alveolar surface tension. And the pressure or the collapsing pressure on a alveolus is proportional to the tension, so the to surface tension, but inversely proportional to the radius. So you understand that a large alveolus has got a low collapsing pressure, whilst the small alveolus will have a high collapsing pressure. So as I mentioned, for the because of the gas exchange, alveoli need to be as small as possible, but obviously small alveoli are not ideal for that tendency to collapse. So how do we solve this problem? Well, this fundamental conflict is solved by the presence of surfactant, as surfactant is able to lower this surface tension on the lining fluid, and by doing so is able to reduce the collapsing pressure. What is the pulmonary surfactant? It's a mixture of amphipathic phospholipids, so molecules that have both hydrophilic and hydrophobic parts. What do they do? Well, they are important because, and not just that, but also phospholipids, protein, and neutral lipid. They are produced by type 2 pneumocyte. What do they do? The surfactant is standing in between the water molecule, and by doing so the surfactant is able to break up the attractive forces in red in here in this cartoon between the liquid molecules. So basically the surfactant is able to reduce the attractive forces between the liquid that is lining the alveoli. And by doing so, they are able to reduce the surface tension. Therefore, they will be able to reduce the surfactant will be able to reduce collapsing pressure of the alveoli, so will increase alveolar stability. And also, the surfactant is important because will increase the lung compliance, so it's easier to breathe. What if there is no or there is not enough surfactant? Well, the alveoli can collapse. And this is something that you may have in premature infants that may suffer from respiratory distress syndrome. Why? Because the pulmonary surfactant is synthesized from the gestational week 24, and before week 24 there is no surfactant, and also the amount of surfactant produced between week 24 and 35 is uncertain. So obviously you understand that premature infants, they might suffer from the respiratory distress syndrome because they have not enough surfactant or they don't produce surfactant, therefore the alveoli tend to collapse. We learned about the function of the lungs and the respiratory system, and I believe that by now you understand that gas exchanges between air and blood occurring in the alveoli are depending on the ventilation. So in the ability of our lung to introduce air in our lung to provide oxygen to our cells, and also to exhale, to get rid of the air, to eliminate carbon dioxide. So ventilation is very important, and obviously ventilation depends on the lung function. And the lung function changes in different physiological and pathological conditions summarized in this cartoon here. So with age, obviously lung function decreases, but also the position of our bodies can affect the lung function, and obviously ethnic group, weight, height, and factors are not listed in here are diseases that obviously can impact on the lung function, as we'll describe later on, and also as you will understand more in future, in next year, when we will talk about therapeutics for lung diseases. So monitoring the lung function or being able to measure the lung function is important. And in order to understand if there are issues with the functionality of our respiratory system. So lung functions are generally measured or referred in terms of volumes and capacities. And changes in lung volume and capacity can be determined using a spirometer. The spirometer allowed you to measure the amount of air you can, well, an individual can move in and out of the lungs during breathing. Now, you will understand more about the spirometry, how it's used when you will complete the online LT-based exercise. More information will be given to you in due course. For now, I'd like you to start appreciating some of the definitions that are important when we think about lung volumes and capacity. So there are static volumes and capacity that are measured in order to measure the lung function. There are 4 standard volumes that you need to memorize, summarized in this cartoon here. The tidal volume, inspiratory reserve volume, expiratory reserve volume, and residual volume. The definition or the description of each of the volume are reported in here. And again, when you will complete the exercise, you will have the ability or the opportunity to actually visualize a typical trace obtained in spirometry to measure these different volumes. But please have a look at the description and try to understand what these different volumes are. What about the capacity? So the ability that define our lung function. There are 4 standard static capacity. Inspiratory capacity, which is a combination between the inspiratory reserve volume, what we can inhale when we take a deep breath, and combined with the tidal volume. The functional residual capacity, which is a combination of expiratory reserve volume and residual volume, basically what we have in our lungs, what is the amount of air that is left in our lungs when we exhale forcefully. But some of that amount of air present in our lung cannot be exhaled completely. And again, that forms the residual volume. But again, you will understand more about this residual volume when you will complete the exercise. For now, please try to remember the description of the different capacity we have been talking about: total lung capacity, vital capacity, inspiratory capacity, and functional residual capacity. The descriptions are here. And also the description of the different volume and capacity are color-coded to help you to memorize or understand their description. Lung function can be measured or evaluated also using dynamic volumes and capacities. These volumes and capacity depend on the rate of airflow, so on the amount of air, usually measured in liter per minute, that can go in and out the airways of an individual. And mostly is derived from vital capacity. As you will understand more this year when we do the exercise, but also next year when we learn about obstructive diseases such as asthma and COPD, the measure of dynamic volumes or capacity is essential for the diagnosis of obstructive lung disease such as asthma. What are they? The dynamic volumes and capacity are the forced vital capacity, so the maximal amount of air that you can exhale after a maximal breath. Now, the difference with vital capacity is that the forced vital capacity is achieved when you force the amount of air that you have in your lung out of your lungs. The forced expiratory volume in 1 second is nothing else than the forced vital capacity, but on the first second. and is the volume of air exhaled during the first second of the forced vital capacity. Again, this is a very important parameter that changes in lung diseases, but particularly in obstructive lung diseases such as asthma. Why do we measure FVC and FEV1? Because abnormality in lung function can be detected or diagnosed if the the FEV1 is less than 80% of the predicted normal value. If the FVC is less than the 80% of the predicted normal value, and if the ratio between if the ratio between FEV1 and FVC is less than 0.7. Now again, you will understand more about these values, how we measure them, what they mean when they are decreased compared to the standard or reference predicted normal value during the exercise. But the take-home message from this is also that spirometry is used to diagnose the presence of diseases and the presence of abnormality, and particularly to detect obstructive and restrictive disorder. The value, the FEV1 and FVC, will change in a different pattern. Obstructive disorder, in the obstructive disorder, for example, the FEV1 FVC ratio is reduced usually, but it's not this pattern is not seen in restrictive disorder. But you will learn more about the difference between restrictive and obstructive disorder and how we diagnose them in next year in PM2C2, when you will learn about therapeutics for lung diseases. To summarize the first couple of videos on respiratory physiology, you learn about the different functions of the respiratory system, you learn about the structure and the histology of the different parts of the conducting zone and respiratory zone. These zones have different functions, and because of that, well, in order to complete the different functions, obviously they have also different structure and different set of cells expressed there. You learn the role of the pulmonary surfactant, and we have been talking about lung volumes and capacity. They are parameters essential to define, measure the lung function, parameters that are changing due to physiological and pathophysiological processes. I introduced here, I have here 3 slides with 3 study questions. Please do complete this question, and model answer will be provided in due course and during the question and answer session for respiratory disorder. For the physiology of respiratory system. And here are some references for you. If you would like to look at the books I've been using to prepare these slides.